This video will take a look at passive transport, or diffusion. Diffusion is the movement from a higher concentration of solutes to a lower concentration of solutes to equalize solute concentration. There are a few different types of diffusion we will be looking at. The first of these is simple diffusion. In simple diffusion, nonpolar, hydrophobic, and small molecules can move across the phospholipid bilayer of the cell membrane. When these molecules diffuse across the cell membrane, they are moving down the concentration gradient, meaning they are evening out the concentration of solutes between the intra and extracellular environments. When solute concentration becomes even in two separate areas, the concentration gradient disappears and an isotonic environment is created. In the diagram shown, simple diffusion can be seen taking place, as the molecules, represented by green dots, diffuse across the phospholipid bilayer in order to even out the solute concentration between the two environments. The next type of diffusion we will be looking at is facilitated diffusion. Facilitated diffusion is passive transport that uses integral membrane proteins to help larger, charged, hydrophilic, and polar molecules across the concentration gradient. The image that was depicted earlier of the cell membrane was only its phospholipid bilayer. Cell membranes also have other key components to their structure. These include cholesterol, which in this diagram is shown in blue, carbohydrates, which are shown in green, and proteins, which are shown in orange. There are two general types of proteins in the cell membrane. The first are peripheral proteins, which can face either the extra or intracellular environments. The second are integral proteins, which span the entire phospholipid bilayer. Two types of integral transport proteins, a channel protein and a gated channel protein, assist in facilitated diffusion. Channel proteins have a passageway that help polar and hydrophilic molecules across the cell membrane. In the diagram depicted, a molecule that otherwise would not have been able to cross the cell membrane is able to get across with the help of a channel protein. The other type of transport protein we are looking at is the gated channel protein, which opens a gate, allowing a molecule to pass through the membrane. Gated channel proteins have a binding site that is specific for a given molecule. In the diagram depicted, a molecule can be seen binding to a gated channel protein that opens its gate on either side in order to let the molecule cross the cell membrane. The final form of diffusion we will be looking at is osmosis, which is the diffusion of water. In osmosis, water diffuses across the cell membrane from the hypotonic environment, where there is more water than a solute, to the hypertonic environment, where there is more of a solute than water, in order to even out the solute concentration. In the diagram, which only shows the phospholipid bilayer component of the cell membrane, water can be seen diffusing across the phospholipid bilayer from the hypotonic environment to the hypertonic environment. Once the water has diffused, the solute concentration is even on both sides of the phospholipid bilayer. When a cell without a cell wall is placed into an aqueous environment, the water diffuses into the molecule, causing it to swell and burst. However, if a cell does have a cell wall, it will still swell with water, but because of the cell wall, will stop intaking water as soon as the cell becomes turgid. In the diagram, a cell without a cell wall can be seen bursting. On the other side of the spectrum, when a cell is placed into a hypertonic solution, it loses water, shrivels up, and dies. A cell with a cell wall has the cell membrane being pulled away from the cell wall when it is placed into a hypertonic environment. This process is called plasmolysis. This video has been presented by John Woodliff-Stanley and Aaron Liedtke.